Hello, everyone. I'm Erik. I'm PPMD's Vice President of Research. Good to see you all in person and not from behind a desk this year. Rachel did mention that for this next session, strategies for mitigating inflammation and fibrosis, we do have a slight change. We won't have an update on Vamorolone, but did remind everyone that Santhera does have the NDA filing for that underway, and we hope to have some updates from them later this year. We're going to hop right into this session. Our first presentation is going to be a pre-recorded talk from Dr. Gil Price with Antisense Therapeutics. And so I'm going to let the AV team take over and start that.
Hello, everyone. I'm Gil Price. I'm on the board of directors at Antisense Therapeutics. I'm sorry that I couldn't be with you today. Antisense is a company located in Melbourne, Australia, and the idea is that, and I'm located in Birmingham, Alabama. The idea is that it would be much easier for me to get to Phoenix than my colleagues in Melbourne. Sadly, that wasn't necessarily the case. My flights yesterday, both flights, I was flying Birmingham to Dallas and Dallas to Phoenix. Both flights, in fact, ended up being canceled. So I apologize for not being with you, but I'm very happy. I'm delighted to be able to share these slides and this presentation with you via Zoom. So today we're going to talk about a variety of different things, but not the least of which there may be some forward-looking statements. And this is our forward-looking statement slide. So as I mentioned, Antisense is based in Melbourne, Australia. They have an advanced stage product pipeline. They're publicly traded, and they've used their drug now in human trials in multiple sclerosis and in Duchenne muscular dystrophy. We're going to be focused on Duchenne and our next steps with Duchenne today. So this won't surprise anyone that one of the keys that we believe is inflammation and how that inflammatory cascade hits muscle cells and muscle fibers. One of the things that we think is important is within that cascade is the management of CD49D cells. And that's what our drug does. Our drug lowers the impact of those inflammatory cells. Let me move on. So what makes it different than, currently we use steroids to manage inflammation. What makes our drug a little different than the standard of care steroid? Well, the fact is that steroids don't do much, if anything at all, to CD49D cells. So in this case, we believe that our drug could be accretive to steroids, but not necessarily needed, because we think that with the response that our drug has demonstrated, that you will have improvements in all of the endpoints that we're familiar with. And we'll talk about a few of those endpoints in a little bit. The drugs like ATL-1102 are oligonucleotide drugs. You've heard of those before. That's the backbone that they rest on. And they're being developed as a once-weekly subcutaneous dose. This is just a busy slide that talks about some of the preclinical studies that we were able to complete in a mouse model. Again, all very successful. This slide just talks a little bit about the clinical trial that we performed, albeit a small clinical trial. There were only 9 boys, but the results were really very positive in terms of lowering CD49D cells and the outcomes that we were looking to improve. Those specific outcomes are pull, grip, pinch, etc. And in each case, there was improvement. So This slide just talks a little bit about the specific core scores and how they improved. I won't read this to you, but you can see this, you can read it on your own. The thing that I'd like to point out on this side, Eugenio Mercuri is well known in the DMD community. And so if you just take a moment to see or to read what he had to say, basically he says the level of improvement achieved was very positive with this drug. I think his endorsement carries a lot of weight and is allowing us to continue our research using this drug. So just in terms of continued research using the drug, here's where we are. We completed this non-ambulatory study. We completed that a while ago, and now we're ready to move into this phase 2B, phase 3 study in Europe. Again, in the non-ambulatory population. And that sometimes is not an area where a lot of drug developers go, but we think that there's plenty of need there and an opportunity for us, just given how effective we believe our drug has shown itself to be. Again, phase 2B3 study, and then we'll have an open label extension into that study. Just a little bit about our regulatory communications. We're on the same page with EMEA. They believe that if we're successful in this phase 2B3 study, that we'll have an approvable package that we can deliver. FDA, on the other hand, has told us that we need to do some additional tox data. We have 6-month tox data. They would like us to have 9 to 12-month tox data. So we'll be working on that in the future as well. So the upcoming program, again, phase 2B3 study, randomized, double-blinded, controlled, 30 clinical trial sites in Europe and the UK with an open label extension. This is sort of the format that we're looking to accomplish. We're going to have 114 participants randomized to either a 25 milligram or 50 milligram or placebo cohort arm. And we will do stratification by steroid use, just so that we can hopefully tell a difference between that variable. The key study population criteria, non-ambulatory as defined by unable to walk 10 meters without assistance, boys aged 10 to 18 years of age, body weight of at least 25 kilograms, and a pull-to entry score of greater than or equal to 2. So the objectives, again, are the ones that you are all very familiar with. We're going to be looking at pull-to, grip, pinch strength, respiratory function, and importantly, quality of life surveys. Are we really helping people improve their quality of life? Well, that's an important aspect for us. We'll be following safety very, very closely and on a real-time basis. Additionally, we're going to be looking for pharmacokinetic data. How well does the drug get into the periphery? And then also lymphocyte and T cell, how does it modulate that CD49D target? So those are our slides, and hopefully this was a good introduction for you to Antisense Therapeutics. Again, being in Australia, we're not maybe as well known as we would certainly like to be in North America, but we're on our way. We're again, making progress with this phase 2B3 study. We feel very good about that, and we're eager to be a part of the community here in North America. If you do have any questions, please feel free to email us at clintrials@antisense.com.au. Don't forget the .au. With that, I will again thank you for your time. I'm grateful, and I wish that I could have been in Phoenix with you. Thank you so much.
Just like to thank Dr. Price for kind of quickly getting that pre-record done for us. and hopefully they can join us next year and share their updates with all of you and take questions from you. Our next speaker is going to be Dr. Larry Markham from Riley Children's, who's the PI on the Fight DMD trial, and share some updates with us on Ifetroban.
Good morning. Make sure my wires are all connected and everything is appropriate. It's a pleasure to be back at the PPMD conference, and it's really nice to actually have the updates that we have been wanting to give for the Fight DMD trial. And so, as was mentioned, I am the site PI. I have no direct financial closures related to this data except for being a principal investigator for the study. And so Ifetroban has been shown to prevent fibrosis and inflammation by blocking thromboxane receptor signaling, and safety has been well established with nearly 1,400 clinical trial participants dosed in nearly 30 clinical studies. These certainly are other disease states other than Duchenne muscular dystrophy, and exceedingly high doses have been used with no adverse events in non-DMD patients and healthy volunteers. It's a dime-sized, once-daily oral medication taken in a fasted state that selectively blocks this receptor. And so you've seen several slides like this over the last day. But Ifetroban, it is believed, blocks the inflammatory response and pro-fibrotic pathways. And so if you look at DMD gene variant, which leads to abnormal or absent dystrophin protein, and that sets off a cascade of events within muscle, some of which is calcium-based. You have degradation of contractile proteins, and then you have intracellular damage, which leads to muscle dysfunction. And so Ifetroban, as you can see on the side there, blocks one aspect of this cascade of events, which the ultimate cascade leads to muscle cell death and then an inflammatory response and fibrosis, which then develops. And all of that leads to increased wall stress, impaired relaxation of the heart, and then decreased contractility or squeeze, leading to clinical heart failure. And so we had some preclinical work which showed that Ifetroban increased survival and cardiac output in two animal models, one in Duchenne, and then one, a limb-girdle muscular dystrophy, and it was well tolerated by the animals. And again, the major outcome of this work was survival of the animals from a cardiovascular perspective. Interestingly enough, after we became involved in this therapy with Duchenne, there was a subsequent paper that came out of, this was not our group, which showed that Ifetroban reduced coronary artery dysfunction in Duchenne animals and improved blood supply to the heart. And so this signaling was enhanced in DMD mice, and then the coronary vasoconstriction was measured with and without Ifetroban. And so there was a reduction in that abnormal coronary vasoconstriction, and so it was felt that that was beneficial. And so with that information, we partnered with Cumberland Pharmaceuticals to consider creating a clinical trial for the use of Ifetroban in Duchenne muscular dystrophy. And this work really came about with consultation with Parent Project and a large number of providers in cardiovascular care in Duchenne to really make this a patient-centered trial that eliminated a lot of things that may not have been necessary, but also included certain aspects of the trial that we felt were important and PPMD felt important to the Duchenne community. And so the study design for the trial is a phase 2 with the primary objective to evaluate efficacy, safety, and pharmacokinetics of Ifetroban in Duchenne. It's a daily oral medication or placebo for 12 months. 2/3 of participants are randomized to active compound, and there are 3 in-person visits in 12 months, which include cardiac MRI, pulmonary function, QMT testing, some quality of life measures, and then 7-day actigraph, which is kind of like a Fitbit for your hand and lower extremity to get a sense of not necessarily muscle strength, but real-world muscle performance. So if an individual can extend their wingspan and reach in their day-to-day life and feed themselves, we feel that's a good measure of what an individual does in the real world. And so then there is an open label extension which is available to all. And you can see on the slide there that there's an early stage cohort which has an LV ejection fraction greater than 45%, and then a late stage cohort which has an ejection fraction of 35 to 45%. And so our key measures are at baseline 6 months and 12 months, include, as we mentioned on the previous slide, one thing that I will commend Cumberland Pharmaceuticals on was the work that they went into the pharmacokinetic evaluation. We did not want participants to spend the night in the hospital or have to deal with that aspect of a clinical trial. And so they worked with a company to pilot a finger stick method for pharmacokinetics. And so the patients are dosed in the clinic, but then observed for a period of time. And then the 8-hour and 24-hour PK studies are actually performed at home, as well as the day 7. The key eligibility and study features, it's all Duchenne boys greater than 7, and that really relates to the ability to tolerate a cardiac MRI without sedation or anesthesia, regardless of mutation type, ambulatory status, and steroid use is eligible. No upper age limit, and standard of care medications which have been approved, deflazacort and all the antisense oligonucleotides, and there's no biopsy or ambulatory testing. So here's an update on the study. I think it was probably 3 or 4 years ago when we were here that I first talked about this trial. So in February 2020, the study was activated. In March, we were paused due to COVID. But then in October, we were able to reopen, and the first patient was enrolled. And so then in May 2022, which is just recently, the early stage cohort has completed enrollment, and our goal is to complete the late stage cohort by the end of 2022. And so here's our enrollment information so far. 37 patients have been screened. 1 patient had a screen failure. 36 were enrolled. 31 in the early stage, 5 in the late stage. 1 individual, the one who enrolled in October, completed treatment and is on open label extension. We had 4 individuals who withdrew prior to ever starting treatment, and 6 withdrew during treatment. And those reasons are: 1 was determined to be ineligible. 1 subject just withdrew consent. There were 2 who had changes in cardiac medications, which made them ineligible, and we had 2 hospitalizations, and I'll talk about those as well. And so our participants who are currently on active treatment, average age of 17 years, but quite a range from 9 to 30 years, and you can see the demographic breakdown there. We are collecting mutation information, and so you can see the breakdown of the different genetic changes. 7 patients were ambulatory and 4 patients have ventilatory support. And you can see the breakdown of other therapies, non-cardiac therapies. So boys are on steroids, ASO therapy, steroids and ASO, or no steroids or ASO. And so safety, in summary, has been well tolerated. There was one interruption due to bruising. 18 patients have reported an event. 3 of those were felt to be possibly related to drug, which included a rash, another type of rash, petechiae, and some bruising. 2 hospitalizations after starting treatment. One was a pneumonia, and one was a stroke, which the deficits were transient. Both were reviewed and felt to be unrelated to treatment, and impact has resolved. In where we are now in the study is we have a data safety monitoring board safety review, which is set to be in July. And then we have an interim analysis for efficacy after 24 subjects complete their 6-month treatment visit, which includes their MRI. These are the sites across the United States, and you can see we tried to get a nice collection of dedicated centers who are spread across the state so that individuals would have access from multiple locations. And so in acknowledging this is really a work that has come together in what I feel with Parent Project's input and talking to patients and families is a relatively straightforward as far as clinical trials go. It's a little bit more than standard of care that you would receive for anyone with significant cardiac involvement. And our goal has been to try to be as inclusive as possible because our focus really is on the cardiomyopathy of Duchenne and trying to devise a therapy within a trial that is helpful for families and patients. And so I want to thank Parent Project. This study is funded by the FDA through Cumberland Pharmaceuticals, and Fight DMD is a foundation in Nashville which has helped with the trial. And that's all I have. Thank you very much.
Thank you, Dr. Markham.
Sure, I'll have a seat. It's a fancy chair.
We do have time for questions. If anyone has any questions for Dr. Markham, here's your opportunity. I can start with one while we're
Sure.
waiting. So the intention here is, so I think you should show that it was standard of care medication. So this would be on top of other cardiac standard of care meds that you would expect families to be on.
Yes, and we didn't have that in the slide, but patients can maintain their current cardiovascular medications, and it doesn't really matter the indication of what they are coming into the trial with, meaning if it's a proactive, preventive medication like an ACE inhibitor that they are on, or if they have had evidence of dysfunction or fibrosis and they are on a medication for treatment of that. The only stipulation is that they are on stable dosing of medication without recent change. And part of the issue for the patients who became ineligible was their treating cardiologist felt that there was an indication to change dosing of medication during the trial, and so that is one of the stipulations.
Great, thank you. And it was great to see that the boys are able to be on other approved therapies. We always talk about combination therapy and the need for that, so it's good to see that happening in practice in the trial, so we can understand.
That was one of the things that was very important to us, because the data in cardiac effect of those therapies is in question, and so to automatically exclude those FDA-approved therapies, we felt was not disingenuous, but it really wasn't going to address the real world of what patients are being treated with.
And then, so you've been working with the FDA on this. I think one of the challenges that we've had in developing trials and testing cardiac therapies in Duchenne is acceptable outcome measures. Have you had any indication with them in terms of what they're interested in as you're looking towards this interim analysis?
Yeah, so this actually, I think, is very interesting, because during my time at Vanderbilt, and you heard John Soslow talk yesterday, and I'm sure he shared data that we started collecting there probably, well, I guess it would have been 10 years ago or more. And so John got an R01 funded grant through the FDA for the first natural history study in Duchenne muscular dystrophy, which is being funded by the FDA. So John, through that grant mechanism, is having ongoing conversations with the FDA about, okay, we're going to collect all of these measures from a cardiac perspective, and then we will take that information that has been collected over time, and then we will come back to the FDA and inform them of, these seem to be the measures that seem most predictive of cardiomyopathy development and progression in Duchenne, and you funded the study, so let's use these measures now to guide future clinical trials. And not that you share too much about grants and things like that, but we're working to take that data and use even further in analysis. So one of the good things about my move to Indiana was there's this really, I don't know if you've heard of it, it's a pretty famous biomedical engineering facility called Purdue University. So they do a lot there with deep learning, artificial intelligence, and things like that. And so we have been engaged with the biomedical engineering department there and are really making some inroads into this data analysis that is more comprehensive than just what is Larry's opinion about Duchenne. This is almost an agnostic approach, is we're just going to take all the variables and look at them together and see what shakes out. And then we can take that to the FDA through this grant to say, okay, here's some very rigorously analyzed cardiac data, and so let's use that to inform future outcome measures.
That's great and desperately needed, so good to hear. I'd like to thank Dr. Markham for sharing the update on Ifetroban in our impromptu interview.
Sure, perfectly fine.
And I think with that, we're going to move on to our next session with our late breaking news. So, thank you. Thanks,